Hey there! We, the students from the Department of Biotechnology and Bioinformatics of Bishop Heber College, are here to give a brief look on the procedure to isolate DNA from human buccal cells by alkaline lysis method. DNA, or deoxyribonucleic acid, is a complex molecule that contains all the information necessary to build an organism. The double helical structure of DNA is simply a polymer of nucleotide. A nucleotide consists of a ribose sugar moiety, phosphate group, and a nitrogenous base. Most amount of DNA is present in the cell nucleus, but a small amount of DNA can be also found in the mitochondria, known as the mitochondrial DNA, as well as in cytoplasm. So without any delay, let's get into the video. The materials that we have used for the experiment are as follows. The test tubes will be used at the end to isolate the buccal cell DNA by addition of chilled ethanol. The centrifugation tube will be helpful in the collection of sample. The 0.9% saline solution will keep the cells from lysing and splitting. 15% SDS here will break open the cell and the nuclear membrane to release the DNA from the nucleus. And of course, the 70% ice-cold ethanol will keep the precipitation of DNA high. Now, about 10 ml of 0.9% saline solution is taken in mouth and swished properly before collecting in a centrifugation tube. About 5 ml of 15% SDS is measured in a measuring cylinder and added in the centrifugation tube along the sides. SDS is an alkaline detergent which aids in lysing of cell and the nuclear membrane during DNA extraction. The laboratory water bath is set at ambient temperature of 65 degrees Celsius to incubate our sample for 10 minutes. Soon after incubation in water bath, our sample in centrifugation tube is spinned at 5000 rpm in a centrifugation machine for 10 minutes. Never forget to balance the centrifuge. Balancing a centrifuge is simple yet critical. Here we have taken water of exactly the same weight and placed on the opposite side of the rotor. After centrifugation, the supernatant is taken in a clean test tube. Equal proportion of 70% chilled ethanol is added along the sides using a dropper. Finally, after a few minutes, we observed white thread-like structures around the solution, which confirms the presence of DNA. I hope you enjoyed watching this video as we enjoyed making it for you. Bye bye. Thank you for watching.